A-Level Biology 9700 Paper 4 Variant 3 May June 2024 Question number 5 and 6 and question number 5 is BRCA2 is a tumor suppressor gene. Its gene product BRCA2 is involved in DNA repair. If the DNA cannot be repaired, BRCA2 has a role in causing the cell to die. BRCA2 is found in the cells of the breast tissues. When a mutation occurs in BRCA2, damaged DNA may not be repaired, and this increases the risk of breast cancer. So when a double-strand piece of DNA breaks, BRCA2 binds to the damaged DNA directly and interacts with the enzyme RAD51 to repair the damage. Repairing DNA prevents other mutations and gene rearrangements from occurring, which could otherwise lead to breast cancer. So double-strand DNA breaks occur naturally during meiosis. So state the event that that is initiated as a result of double-strand breaks during meiosis. And when meiosis is started, so at that time, in prophase 1, the homologous chromosomes come close to each other, and at that time the crossing over occur. So we will write that crossing over, in which the chromosomes come close together, the homologous chromosomes come close together and exchange their parts. Now, the next question is, scientists have identified hundreds of mutations in BRCA2, but not all of these mutations will increase the risk of cancer. One specific mutation in BRCA2, known as 999DEL5, is found in 0.6% of the general global population. Iceland is an island country in the North Atlantic Ocean. The ancestors of most of the current populations are people who arrived to settle in Iceland in AD 874. In the Icelandic population, mutation 999DEL5 is the cause of 7 to 8% of breast cancer cases in the women and 40% of the breast cancers in men. This is the much higher than the percentage of breast cancer cases in the general global population. Suggest and explain how mutations 999DEL5 accounts for a very high percentage of breast cancer cases in Ireland compared with the general global populations. So we have to give suggestions about this, that how mutations account for a very high percentage of breast cancer in Iceland compared with the general global populations. So, the mutations are the sudden change in the sequence of the DNA or in the number of the DNA. And in this case, the mutations is a, you can say it shows a deletion mutation, which means a specific DNA sequence of 999del5 base pairs has been removed from a particular gene. So a gene deletion can occur in a population and contribute to the reduced genetic diversity and characteristic of a founder effect. So the founder effect is about the reduced genetic diversity due to small population. So if the deletion occur in a tumor suppression cell, it could lead to a loss of function of gene. With the loss of function, cell growth may become uncontrolled, increasing the risk of cancer development. So in this way, we can say that how mutation 999del5 accounts for a very high percentage of breast cancer cases. So in this way, I would like to say, uh, after this explanation, I am going to write this explanation in the given space. And here I will start with, that is, that it can be an, you can say, It shows it shows a deletion mutation which means that a specific DNA sequence of triple nine del 5 base pairs has been removed from a particular gene. So a particular gene, a gene deletion can occur in a population. You can say a small population and can reduce genetic diversity. Diversity characteristic and a characteristics that of founder effect. And you know the founder effect is about the reduced genetic diversity and small population due to small population size. And then you can say the deletion can also occur if the deletion occur in a tumor suppressor gene. It could lead to a loss of function. Cell growth may become uncontrolled, increasing the risk of cancer development. So in this way, I would like to say we can give these three examples as deletion mutation in which DNA sequence has been removed. Other founder effect, we can refer the founder effect due to which a smaller population have reduced the genetic diversity. And the third, the deletion of the tumor suppressor genes due to which uncontrolled uh, you can say uncontrolled um, cell division increases the risk of cancer development. So it was all about this question. Now, next question is, one of these global genetic screening programs for breast cancers identifying people with mutations in BRCA2 outline the advantages of genetic screening for mutations in BRCA2. So, students, you know that uh, genetic screening programs are, you can say, very helpful in the case that the, we can identify the people who are suffering with this disease, uh, or you can say it can also uh, give health benefit to the people, and the people come to know whether they are on which stage of this disease. So I would like to write the answer, that is the advantages. Many advantages are in the way. So we can say, if the person who is positive with the disease, with this and he come to know that he is suffering uh, with breast cancer, then he can manage his treatment. So I'm going to write all this in the way, that is, if the person, many advantages, one of them is, if the person is positive, then he or she will be able to catch the disease at its early stage, and can start treatment in this way. Death rate can be reduced. On the other hand, if test is negative, then the person will be will be tension free. So in this way, uh, there is an advantage of genetic screening for mutations. And the next question is, suggest one advantage to a country of a genetic screening program for breast cancer that screens for specific mutations in BRCA2 in the population. So here there are many advantages, but I would like to, we have to write only one advantage. And you can say, by identification of the individuals with harmful mutations in these genes, the programs can pinpoint those at significantly increasing risk of developing breast cancer. So these high risk individuals can then be offered more frequent and intensive screening. So in this way, I would like to say that we are going to write only one, by identifying with harmful mutations. We can find out with this the it can be find out the person who are suffering with these harmful mutations. genes in those persons program can pinpoint. Those at high risk. So their treatment can be started at early stages. So if they are identified, so the best method is, or you can say, this is the advantage, that is the identification of those people who are at the risk. So they can be pinpointed, and in this way, their treatment can be started. And the next question is uh, lipoclain 2. Lipoclain 2 is a cancer promoting gene, oncogene. When LCN2 is expressed, it can result in breast cancer. Research is being carried out to see if gene editing of LCN2 could be used to treat breast cancer. Gene editing was used to treat human cancer cells that had been implanted into mice to form a tumor. The treatment stopped LCN2 from being expressed in the cancer cells and resulted in a significant reduction in the growth of the tumor. There was no negative effect in the normal tissues. Now suggest how DNA editing stopped LCN2 from being expressed in cancer cells. So we are going to write that the how DNA editing. So we will discuss about DNA editing. So in this way, the DNA with the DNA editing, you can say this system of DNA editing is used uh, to, you can say, in which the DNA is cut, and it can be by the which, first of all, they identify the uh, target DNA. Then the DNA can be removed, it can be deleted, it can be replaced. So in this way, first of all, the identification of the LCN2 and gene, which involves, and then the understanding the gene sequence and its you can say its regulatory element. So complete deletion of the gene in some cases, and the entire LCN2 gene can be deleted, or preventing an expression of this gene can be reduced by replacing muted sections of the DNA with correct sequence. So in this way, there are different uh, ways by which the DNA gene editing can be done. So I will start with the gene editing is a system is used to target DNA is cut, be replaced, deleted. So that could be deleted, replaced, and in this way, first of all, identification is identification ELC and 2 is necessary. Then gene sequence, to know the gene sequence and its regulatory element. Complete deletion of the gene and in some cases the entire LC and 2 gene can be deleted, preventing expression, its expression. Or by replacing muted section of DNA with correct sequence. So, uh, first of all, I will explain it again that first of all, the gene editing is necessary, and the gene editing to we can insert a sequence of faulted DNA, we can replace it, we can delete it. So in this way, first of all, identify the wrong sequence and then it can be replaced. So sometimes the sequence can be added which can stop the gene expression, wrong gene expression, or sometimes some part of it can be replaced due to which the cell growth can be reduced. Now the next question is, suggest why the expression of only L, C, and 2 was affected. Okay. Suggest why only, because only uh, target sites on DNA only found in LC and 2. So that was the reason that only LCN2 was affected. Now the next question. In plants, stomata open and close in response to changes in the environmental conditions. Explain why stomata need to open and close according to environmental conditions. So, students, the stomatas are the small pores uh, which are meant for gaseous exchange and for transpiration as well. So, stomatas close in the morning and when there is sunlight. At that time, the process of photosynthesis takes place. For photosynthesis, the plants take in the carbon dioxide, and as a result of photosynthesis, the oxygen is produced. So the oxygen is used for respiration, and remaining oxygen is released out through stomata. So in this way, the other case is transpiration. When water is evaporated through stomata, extra water is evaporated through stomata, and on the other hand, these stomatas are closed when there is no photosynthesis. So at night time, these stomatas are closed to reduce the water release out. So in this way, the plant get dehydrated. Plant get hydrated if more water is evaporated. So the same is there due to the transpiration takes place. So the as a result of transpiration, sometimes when less water is available and dry or sunlight is more, then at that time more water can be evaporated and water and plant get hydrated. So avoid all these conditions. These stomatas are closed, or when there is no photosynthesis, stomatas are closed. So I would like to write this. First of all, that is stomatas open in when photosynthesis takes place. We take carbon dioxide and remove extra oxygen. During transpiration, stomatas also open to evaporate water, and then stomata are closed at night when there is no photosynthesis and to reduce the rate of evaporation, to protect from dehydration. Next question is, describe the mechanism occurring in guard cells that leads to the opening of the stomata. Well, when the students, the guard cells are open when the process of photosynthesis takes place. So, how do they open? Uh, when there is sunlight, so this process takes place. Photosynthesis takes place. So, at that time, because plants that has to get the carbon dioxide, they have to release the oxygen. So, the stomata should be open. So this process takes place as when light strikes the leaf. It triggers a series of biochemical reactions in the guard cells. So photosynthesis in the guard cells produces ATP and NADP. So the energy from ATP is used to actively pump potassium ions into the guard cells, because already protons, or you can say the hydrogen ions, are moved out of the leaves through uh, these hydrogen pump, and the this increases the solute concentration or when potassium ions enter. So it increases the solute concentration in the guard cells, due to which water moves into the guard cells by the osmosis. So this influx of water increases the water potential and guard cells become turgid, and it causes them to open. So, in this way, the stomatas are open. So I will write this at the time of photosynthesis, when light tea leaves triggers a series of biochemical reactions in the guard cells. Because chlorophyll absorbs sunlight and photolysis occurs, which release ions the energy from ATP is produced and ATP is used to actively pump potassium ions into the guard cell. Because due to outgoing H positive ion, the negative has been increased. Exactly. When ions enters, then concentration is increased and due to less water pressure, water enters into guard cells by osmosis, due to which guard cells become turgid, which causes them to open. So student, it was all about question number 5 and 6. Please like, share and subscribe my channel. Thanks for watching.